Hello, my name is Marcin Joachimiak. I'm a staff researcher at Lawrence Berkeley National Laboratory. And today I'd like to tell you about our recent work on KG-Microbe, a reference knowledge graph and platform for harmonized microbial information. KG-Microbe leverages a knowledge graph construction pipeline and template that our group has developed in the last year. This covers standard cases for data ingestion, integration with named entity recognition tools, tools for interacting with ontologies, as well as KG construction software for merging and summarizing the data sources. I also, throughout this poster, I provide links for the resources, appropriate resources. The data sources that we prioritize for KG-Microbe follow along the KG Hub principle. So taking a set of ontologies that will allow to interlink disparate data types. In this case, we started with a microbial traits table published by Madden et al., which actually includes a number of different entities. And so this was a good test of our NER tool integration using, again, the provided ontologies as dictionaries. We're able to map a number of features from the microbial trait table, including taxa, chemicals, environmental terms, and molecular functions. And these are standardized in the knowledge graph setting and also correspond to a build process with releases. So we prioritize our work using GitHub and tickets, including requests for users. Mostly these are data published in different papers, as well as some online resources. As part of the KG-Microbe infrastructure supported by KG Hub, there is an auto-generated documentation resource provided with this link. And so this could be useful if people wanted to clone or fork this repo and kind of go down their own path of appending to it, maybe splitting into biomedical or environmental applications. We also have a public release directory structure at the provided link. And here we include builds, results from builds and deployments of the knowledge graph, which happen monthly via GitHub integration with Jenkins. The directory structure includes both the raw and transformed individual data sources, the merged knowledge graph summary statistics, as well as the knowledge graph files themselves. The KG Hub also provides a knowledge graph dashboard, which is a high-level summary of the knowledge graph itself. And because we use the BioLink model to model the entities and relations in the graph, we can use the categories from the BioLink model to provide some very useful summaries. So this actually can be used to also inspect the knowledge graph and track its state, to identify data types or data sources that are underrepresented, and also to check whether normalization has happened in appropriate ways. We can convert the graph data to a table to enable other machine learning analysis. However, the information in KG-Microbe and a lot of similar knowledge graph resources is quite sparse, so the graph structure is a more appropriate data object. However, we can create the table, and there's a little histogram here showing the feature frequency of the columns generated from the knowledge graph, KG-Microbe knowledge graph in its table version, and you can see that most of the features are quite sparse, and so only apply to a small subset of the over 12,000 taxa represented in this data set. In terms of the taxa, we also used a ontology interaction tool called Robot to trim the full NCBI taxonomy and to arrive at a subset of the taxonomy relevant to microbes. And so this allowed to both scale down the immense size of the NCBI taxonomy and also craft it to specific needs in a knowledge graph setting, which also set a precedent and will be reusable in KG Hub. Now, one interesting and kind of exciting use of knowledge graphs are graph embeddings. And we perform graph embeddings using the Embedding package provided at the GitHub link, using a node-to-vec implementation that really only use the structure of the graph, so the nodes and their linkages. There's additional information that other algorithms could make use of, including edge types and node and edge properties. And so here I'm showing two t-SNE dimensionality reductions of the graph embeddings, both for nodes and edges, left and right, colored by their corresponding BioLink model types. And so you can see that this reveals some interesting structure and relationships of the data. Mainly the thing we are looking for here is no kind of circular balls of data which would correspond to low dimensional embeddings with low information. And so the goal, so this can actually be used as a diagnostic in the KG construction process to identify pieces of data, substantive data that either are poorly linked or do not provide any novel information. And again, the node and edge categories are provided from the BioLink model, which is actually a very nice, useful aspect of this whole KG construction process. And finally, one demonstration of the application of the KG-Microbe through the graph embeddings is a predictive model to predict microbial shapes from the data in this table, which I should note does not include any sequence or genomic information. And so using the data sources shown in the middle panel, so the ontologies interlink with the microbial trait table for chemicals, functions, the taxonomy itself, and in environmental terms, we're actually able to build quite predictive models of microbial shapes. And so here I'm showing both the feature importance plot, the confusion matrix, and a performance table. We used CatBoost, the CatBoost package, to build these classifiers using the graph embeddings as a direct input, so as input features. CatBoost uses a gradient boosted decision tree methods to construct the classifier. And as part of this process, we split the data into test and training, generate additional negative associations via some random mismatch methods, and we evaluate the performance on 20% withheld data. And so the results I'm showing here, which in most cases near perfection, except for a few cases which have lower representation, so kind of rare classes, which are a common issue, we can perform quite well in predicting microbial shape, which I think traditionally has been something difficult to do and would probably rely more on sequence information or even images from microscopy. So in this case, we didn't have to rely on either of those expensive and difficult to obtain data types. So we think that KG-Microbe will be an important resource for microbiology and analysis, and we plan to develop it further and interact with the community to both gather feedback and learn about applications. Thank you.